So, welcome to all of you in today's session. Yesterday we have studied in my previous lecture we have studied character properties of enzyme, and then in today's lecture we are going to discuss very interesting topic, that is how enzyme basically works, or you can say kinetics of enzyme. You see that the basic question is that what enzyme kinetics is actually. So, the enzyme kinetics is the study of analyzing the reaction rate and its variation under different experimental conditions. So, this method is very important for understanding the mechanism of enzymes action, and very significant that to determine the feasibility of the reaction. So, here that in the slide you see that a graph has been plotted between the concentration of the substrate and action velocity, showing the sort of saturation curve here. So, basically the velocity of enzyme catalyzed reaction is dependent on the concentration of substrate, but what happens initially, you see that at low substrate concentration the initial velocity V0 increases almost linearly with the substrate concentration. You see that here the velocity increases linearly with the substrate concentration. As you see that from this graph, as the concentration of substrate increases further, generally the reaction reaches a maximum velocity and does not increase beyond this point. So, at high substrate concentration, generally V0 becomes virtually independent of substrate concentration and generally approaches a maximum limit, which is known as Vmax. You see here there is a saturation, so Vmax is achieved, and generally this shows the saturation effect. But think of the condition that low substrate concentrations, velocity is generally proportional to substrate concentration, indicating the first order kinetics. However, at high substrate concentrations, velocity becomes virtually independent of the substrate concentration, leading to zero order rate of reaction or zero order kinetics. So, you should note at this point that what is the main difference between first order kinetics and zero order kinetics. So, this is very important to note the significant difference between these two types of kinetics. Now, you see that basically Michaelis and Menten proposed a very straightforward model that explain the most important aspect of enzyme catalyzed reactions. Basically, in this model, the enzyme combines reversibly with the substrate to form enzyme substrate complex, which then produces the product restoring the free enzyme. So, in this reaction, generally a single substrate has been taken to explain the Michaelis-Menten equation, and here you see that S represents the substrate, E represents the enzyme, and ES basically represents the enzyme substrate complex, P represents the product, and K1 here is the rate for the formation of enzyme substrate complex from the enzyme and substrate, and K minus 1 basically is the rate constant for the reverse reaction of the enzyme substrate complex. K2, you see that here K2 is the rate constant for the conversion of enzyme substrate complex to product and the subsequent release of product from the enzyme. So, as per this Michaelis-Menten concept, we can measure how fast an enzymatic reaction occurs with different amounts of substrate. You see that the rate of reaction depends upon the concentration of substrate. Here on the x-axis substrate concentration has been taken, on y-axis rate of reaction has been taken. So, when condition is that when there is not much substrate around, generally the reaction is speed up as we speed up, and once we add more substrate, almost like climbing stairs on one step at a time. But once we have a lot of substrate, the reaction hits its top speed called generally Vmax, and adding more substrate doesn't make it to go the reaction rate faster. It means it does not impact the rate of reaction. So now then Michaelis-Menten constant reveals what? Question arises, this Michaelis-Menten equation, Michaelis-Menten constant reveals what? So basically this is Michaelis-Menten constant is the concentration of substrate at which the enzyme reaction is happening at half its maximum speeds. It's like basically finding the sweet spot for the reaction to work efficiently, and you see that this Km can be calculated by adding together two another constants, namely K minus 1 and K2, and dividing by another constant that is K1. So it's generally Michaelis-Menten constant is measured in terms of moles of substrate per liter, and generally a lower Km value means that enzyme really likes that substrate and grab the substrate very tightly. Now come what Michaelis-Menten equation is basically. So Michaelis-Menten equation actually came up with a sort of handy mathematical formula to explain basically how enzymes, substrate and reaction speeds are related together. And you see that in this equation basically V0 is equal to Vmax into concentration of substrate divided by Michaelis-Menten constant plus concentration of substrate. So what this basically this is V0, please note it here, it's V0, and what V0 represents basically it's initial reaction velocity, and Vmax is the maximum reaction velocity, which is how fast the reaction can go when the enzyme is fully loaded with substrate. Km just could be calculated, just I mentioned that it's equal to K minus 1 plus K2 upon K1, and S stands for concentration of substrate. So in a very simple term, you see that this equation talks about how fast the reaction is going to be depending on how much substrate is around. So basically it helps us to understand the relationship between enzyme, substrate and how quickly they react together. So there are different assumptions are made basically in driving the Michaelis-Menten rate equation. First is the relative concentration of enzyme and substrate. How relative concentration of enzyme and substrate impact the rate of reaction. So the substrate concentration, that is S, is significantly higher than enzyme concentration. This is the assumption. So ensuring that only a small percentage of the total substrate is bound by the enzyme at any given time. Second, steady state assumption, the concentration of enzyme substrate complex remain constant over time. This is known as steady state assumption, meaning that the rate of formation of enzyme substrate complex is equal to the breakdown of enzyme substrate complex. The third assumption, once there is a very little accumulation of product, so the formation of enzyme substrate complex from enzyme plus product is generally negligible. So you see that how this Michaelis-Menten equation tells so many aspects of enzyme catalyzed reactions. Now, what are the conclusions could be drawn from this Michaelis-Menten equation? So, what are the characteristics of Michaelis-Menten constant? First, I will discuss. So, the Michaelis-Menten constant is very specific to an enzyme substrate pair and indicates the enzyme's affinity for that substrate. It's basically the substrate concentration at which the reaction rate is half of Vmax, as you see, half of Vmax here, and remains constant regardless of the enzyme concentration. So, small km indicates what? Small km indicates what? You see that here km is small. So, it indicates basically high substrate affinity. Small km indicates what? High substrate affinity, or you can say in other words that is a low substrate concentration is sufficient to reach half of Vmax. You see in this figure, this is the first condition I am mentioning, where Km is small, and then in this enzyme 2, you see that large Km is there. So what does it indicate? It indicates basically large Km indicates how substrate affinity and low substrate affinity, that is high substrate concentration is generally required to reach half of Vmax. So then how this equation reveals the relationship that between velocity enzyme concentration. So generally you see that reaction rate is directly proportional to enzyme concentration at all substrate concentration. For example, if the enzyme concentration is halved, the initial rate of reaction, that is V0, as well as that of Vmax are reduced to half of that original. So now what is the impact on order of reaction? This is very interesting to share. Suppose when S is much less than substrate concentration, or you can is less, or you can say at low substrate concentration than the Km, then what will be the fate of reaction? Try to understand this aspect because this aspect is very crucial and generally asked in number of competitive examinations. So try to understand this aspect. What will be the fate of reaction? Suppose when low substrate concentration, or you can say substrate concentration is less than Km, then reaction velocity is approximately proportional to the substrate concentration. So this is known as first order kinetics. This is known as first order kinetics. Here I mentioned. Then second condition, when suppose concentration of substrate is much greater than this condition, at high substrate concentration than the Km, or you can say when substrate concentration is greater than Km, so velocity remains constant, Vmax remains constant regardless of substrate concentration. So this is known as zero order kinetics. So this indicates basically enzyme saturation with substrate. So one is the zero order kinetics and one is the first order kinetics, and this could be revealed by Michaelis-Menten equation. Now this is very important aspect, catalytic constant of an enzyme. So we will go into the details of what catalytic constant is basically. So catalytic constant of enzyme is generally often denoted by a constant K catalyzed, you see here. This is also referred to as turnover number and represents the maximum number of substrate molecule converted to product by a single enzyme molecule in a unit time when the enzyme is fully saturated with substrate. In other words, you can say that it basically this catalytic constant quantifies the efficiency of an enzyme in catalyzing a specific reaction. So this could be expressed, this catalytic constant could be calculated by calculating Vmax upon E, and where you see that Vmax is the maximum reaction velocity achieved when the enzyme is fully saturated with substrate, and E is the concentration of enzyme. So basically this catalytic constant provides important insights into enzyme kinetics and also catalytic efficiency of enzymes. So what does it indicate? This catalytic constant, basically a high catalytic constant value indicates that the enzyme can convert substrate molecule into product at a rapid rate, whereas low K value or catalytic value suggests that lower catalytic activity of enzyme. So it's very important to note that the catalytic constant is a characteristic property of each enzyme and is specific to particular reaction it catalyzes. This is influenced by various factors including temperature, pH, substrate concentration, and presence of inhibitors or activator. For example, I would like to share with you one interesting example of catalase. So this enzyme catalase has the highest turnover number recorded so far. So each enzyme molecule can break down 40 million hydrogen peroxide molecule per second. So opposite to it, lysozyme is another interesting example. It takes generally 2 seconds to cleave a glycosidic bond in peptidoglycan present in the cell wall. Just see how catalytic constant is very low for this enzyme lysozyme. One is highest catalytic constant and one is lowest catalytic constant. So these are the very interesting example you must remember. Now another one is a specificity constant of an enzyme. What this specificity constant indicates, generally it could be calculated by calculating specificity constant upon Michaelis-Menten constant, and generally it's a measure of the efficiency and specificity of enzyme catalyzed reaction. Basically this constant combines two important parameters, that is catalytic constant, which represents the turnover number or the rate, you can say, which enzyme converts substrate into product when it is generally fully saturated, which we just discussed before, and the Michaelis-Menten constant, which generally indicates the affinity of enzyme for its substrate. So this could be mathematically calculated. Enzyme specificity constant could be calculated by the simple equation catalytic constant upon Michaelis-Menten constant, just where Km I have already discussed is the catalytic constant representing the maximum rate of enzymatic reaction, and Km is the Michaelis-Menten constant representing the substrate concentration at which the reaction rate is half of Vmax. You see that the specificity constant provides basically a quantitative measure of how well an enzyme performs its catalytic function relative to its affinity for the substrate. So generally a high specificity constant indicates that an enzyme has a high catalytic efficiency, meaning it can efficiently convert substrate to product even at low substrate concentration. Opposite to it, a lower specificity constant implies lower efficiency and specificity in converting substrate to product. And basically this specificity constant is particularly very useful for comparing different enzymes catalyzing the same reaction or for evaluating the effectiveness of enzyme variants engineered for improved catalytic performance or altered substrate specificity. So, just understanding the specificity constant is essential in enzyme kinetics and in revealing the mechanisms underlying as enzyme substrate interactions. It also has a very practical application in various fields like biotechnology, drug discovery, and for optimizing the performance of enzymes, uh, uh, particularly for various targeted therapies in medical sciences. Now, Lineweaver Burk plot I would like to share in relation to Michaelis-Menten equation. So, generally what happens that it is quite difficult to determine Vmax and Km directly from a plot of um, velocity against the substrate concentration. So, it could be make easier by rearranging the Michaelis-Menten equation in different ways to give a convenient graphical representation. So, a graph known as Lineweaver Burk plot or double reciprocal plot. You should also remember this Lineweaver Burk plot is also known as double reciprocal plot, and this could be made by plotting reciprocal of the velocity, that is 1 upon v here, 1 upon v0 against the reciprocal of the substrate concentration 1 upon s. So, this special plots turns the Michaelis-Menten equation into a straight line equation. So, how this Michaelis-Menten this Lineweaver Burk plot convert the equation Michaelis-Menten equation 1 upon v0 is equal to Km upon Vmax into 1 upon S plus 1 upon Vmax. So, with the Lineweaver Burk plot we can easily find Km and Vmax, and the slope of the line, basically this slope of the line just give us the Km value, while the intercept gives the Vmax as shown in this graph. So, this Lineweaver Burk plot has many advantages. Generally, it helps us to estimate Km and Vmax even if our data is bit you can say uneven, and it serves a great tool you can say for comparing different enzyme kinetics data all in one graph. Now, Hanes-Woolf plot is another type of plot. It shows the graphical representation of enzyme kinetics and generally provides a linear transformation of Michaelis-Menten equation. So, this Hanes-Woolf plot involves plotting the ratio of basically initial substrate concentration against the initial substrate concentration. So, basically the Hanes-Woolf plot is based on rearrangement of the Michaelis-Menten equation. It is generally considered more accurate than Lineweaver Burk plot for the determination of basically kinetic parameters, and it aids in understanding the enzyme kinetics by providing a clear visualization of how different factors such as inhibitors, enzyme substrate interaction and reaction rates alter, and the graph and equation you just see in this slide, where S upon V0 has been taken on y axis and substrate concentration on the x axis. Now, what is the significance of Michaelis-Menten constant and Vmax? So, those basically both Km and Vmax are very important parameter in enzyme kinetics and provides valuable insights into the activity of enzymes and their regulation. So, generally just I mentioned that this provides Km provides information about the affinity of an enzyme for its substrate with low Km value indicating generally high affinity of the enzyme for its substrate, whereas a high Km value indicates a low affinity. So the value of Km is very useful in basically understanding how changes in substrate concentration affect the reaction of rate of reaction. Vmax, on the other hand, you see that it's a very interesting parameter which represents the maximum rate at which an enzyme can catalyze a particular reaction. It is the basically maximum velocity achieved by the enzyme when the substrate concentration is high enough to saturate the enzyme. And the value of Vmax is dependent on the number of active enzyme molecules present and generally does not change with the change in substrate concentration. So what is the use of this Vmax? So the value of Vmax is the very useful in understanding how changes in the number of active enzyme molecule can affect the rate of reaction. So both Km and Vmax are very much useful to characterize the behavior of enzymes in various industrial and biotechnological applications. For example, enzymes with high Vmax value are preferred in biocatalytic process because they can catalyze reaction at a very faster rate, whereas the enzyme with low Km values are preferred in processes where substrate specificity is very important, such as in the synthesis of specific chemical compounds. Now, we have in our previous part of the lecture, we have focused on enzyme catalysis by taking only single substrate. So, but you see that in many reactions, generally multiple substrates are involved. For example, alcohol dehydrogenase catalyzes the reaction where ethanol and NAD produces acetaldehyde, NADH and a hydrogen ion. So this enzyme basically binds with NAD and the substrate to be oxidized. While the principle established for single substrate system can be extended to multi-substrate system, the complexity further increases. Generally, these multi-substrate system reaction involve the transfer of a particular functional group from one substrate to another, and then the enzyme can bind, for example, two substrate in various manners, A and B. Here it has been shown. So there are basically two categories of substrate binding with enzyme. One is sequential and other one is non-sequential mechanism. So first I will discuss sequential mechanism. In certain reactions, both substrate that you see A and B must bind to the enzyme before the products are released. So one is this category of sequential processes could be further divided into ordered sequential mechanism and random sequential mechanism. Now what is ordered sequential mechanism? So basically in this mechanism, the binding of one substrate is necessary before the second substrate can bind, and the horizontal lines basically are the enzyme, and this one is enzyme substrate complex, then second substrate bind, and then product is formed. And so basically this ordered sequential mechanism encompassing both forward and reverse reaction, and this mechanism is generally observed in oxidation of substrate by NAD. Now random sequential mechanism, this is a condition where the binding of substrate and the release of the product do not occur in a predetermined obligatory order and bind to the enzyme randomly. Here you see that it has been shown. Basically, the very interesting example is the phosphorylation of glucose by ATP to form glucose 6-phosphate and particularly this reaction is catalyzed by hexokinase, generally initiating the first step of glycolysis, is very interesting example of random sequential mechanism. Now, non-sequential, or you can say this is also known as ping pong mechanism. Non-sequential or ping pong mechanism. This process is termed as ping pong mechanism to highlight the generally bouncing of enzyme between the two states. You see that bouncing of enzyme in two states, E and E plus. So basically these are the two states of enzyme, and enzyme bounds in these two states. That's why this mechanism is known as ping pong mechanism. So you see that first substrate A generally binds to the enzyme and undergoes a reaction, resulting in the formation of chemically modified form of E here, along with the product here. Then the second substrate B reacts with E modified enzyme, restoring the original enzyme and the product Q. So generally such kind of reaction following this pattern, they are also termed as double displacement reactions, and chymotrypsin action basically fit this type of non-sequential or ping pong mechanism. So thank you very much for patiently listening this very important aspect of enzyme kinetics. Thank you very much.